Okay, welcome back to our biochemistry review videos. We're going to do a little review of lipids now and what they are and what are some molecular structures that what they look like. Well, first off, they are another organic molecule. Earlier on, we talked about carbohydrates being organic, and these are organic too. They have the same atoms, the same elements that make this up: carbon, hydrogen, and oxygen. So, being that carbon and hydrogen make up organic molecules, this is an organic molecule. Okay. Typically found in fats, oils, waxes, cholesterol, and they're composed of one glycerol and 3 fatty acids. That'll make up one molecule of a lipid. Those are your subunits, or basically break down fat into these molecules here. Functions: long-term storage of energy, insulation, makes up a good amount of the plasma membranes. Another word for plasma membrane is cell membrane. And we're going to talk about phospholipids as being part of the cell membrane later on. The cushion organs also as well. Okay, so that's some of the obvious ones. Okay, so now what we're looking at here is actually a glycerol. And glycerol is easily being found by looking at the 3 carbon backbones surrounded by the hydrogens and the hydroxide ions. And right here we have what's called a fatty acid. You can tell it's a fatty acid. It's a long chain of hydrocarbons, and it has this carboxyl group right here. It's a hydroxide. But this carboxyl group definitely is on the end. Let's look at that in a second. Okay, so what do we need to do to make a lipid? Well, basically we have to combine this fatty acid and attach it to the glycerol. You might be thinking how to do that already. And it's not too bad, but let's try to scroll down so you can see. Okay, here's our glycerol. What we need to do is take out that OH again, and that hydroxide, we should say, and we have to take out a hydrogen, and from there now this oxygen is ready to bond with this carbon. So we can bring that down. Try scroll down, and let's see. Okay, so hopefully you were able to see all that, and you saw that the fatty acid was able to bond with the glycerol. Okay, and what did we take out? Water. So again, we took out water, so it was dehydration, and we built a bigger molecule, so that's synthesis. Okay, and if power TV, I already did the other 2 fatty acids. If we have 3 fatty acids bonded to a glycerol, we consider that a lipid. Okay, well, I do appreciate you tuning in. Hopefully you learned something, and you will apply this to your test on Friday. Okay, thank you, and bye. Okay, welcome back to our next video with our biochemical molecules. Sometimes we call them our macromolecules. Right now we're looking at an amino acid, and the way we can tell it's an amino acid is that it has this nitrogen group here. It's called the amino group or the amine group, and it's NH2. You can see that. It has 2 carbons here, one of them part of this carboxyl group. And then there's another hydrogen. That's pretty standard, these parts of the amino acid. But this part right here is what we're worried about. It's the R group or side chain. And that's the variable group. And that's what really determines what amino acid has what characteristics. Take a look at a couple of more specific ones. So here's again our general structure of it with our R group. And the R group down here, that's different for every amino acid. So this one's CH3. This one over here, we can look at, has a little bit bigger of an R group, CH3, and sulfur and hydrogen So these are two different amino acids that we can look at. Okay. We can actually talk about forming bonds between amino acids, and we can see that we do have to start to take something out again, and what we're going to take out is this hydroxide from this carbon over here, this carboxyl group, and take out the hydrogen from that amino group, or amine group, and we can see how this is going to fit together. We kind of have to do a little extra rearranging here, but we didn't really see that. So basically, we're going to take this amino acid now. It's capable of bonding over there. It's not going to fit neatly right now, but we can kind of shape it up a little bit, and we might need to move water out a little bit. Sorry about all this. And we even have to kind of move some stuff around a little bit. So hydrogen's sort of in a way, but it's still bonded there. And maybe we can move it a little bit closer. Not always an easy task here. And look at how this carbon is going to form a bond with nitrogen. And that bond right there is a special bond. We call that a peptide bond. Okay, so we have right now here's our one amino acid over here with that CH3 side group, and we have another amino acid over here, which again that has that little bit longer side chain. So we have two amino acids bonded together with a peptide bond, and that's going to give us our dipeptide. Sorry, it's late today. So, and what did we take out? Water. So again, this is dehydration. Take out water. Synthesis. We're putting two molecules together. So this makes our dipeptide. Now, if we did that, don't always like to draw all those carbons and hydrogens everywhere. So a lot of times we just draw circles to represent our amino acids on these polypeptides. So this right here, okay, again, amino acid, amino acid, amino acid, multiple amino acids bond together with these peptide bonds. That's going to give us our polypeptide. And that's also going to give us our primary structure of proteins. Then what we get is, due to interactions between R groups and hydrogen bonding and polarity and all that stuff, you get a certain folding going on due to the interactions, and that's called our secondary structure. Continue to fold up into more of a three-dimensional shape, which is going to be our tertiary structure, and finally our most complex protein in our shape is going to be our quaternary structure. And a lot of times we think about with enzymes having an active site. Basically, an active site is a certain region within our protein, within our enzyme, that actually has an interaction between substrates. And substrates will come in and bond with our enzyme, and it'll allow certain chemical reactions to occur much faster because of that special place and shape on that enzyme and interactions between it. Okay, I think that's going to conclude our discussion about proteins for now. I hope you learned something and that you can use this in your studies. All right, thank you very much.